So I'm going to be talking about work that we've been doing to map the E. coli functional proteome using thermoproteome profiling. So let me start by explaining what thermoproteome profiling, or TPP, is. So the principle behind it is very simple. When you heat up proteins, they denature and aggregate. So if we monitor the remaining soluble fraction as a function of temperature, we get something that resembles a melting curve. Now with TPP, we do this in living cells and at the proteome level, which means that we get one of these melting curves for every protein of the proteome. And the reason why we care about these melting curves is that protein thermal stability is affected by protein interactions. For example, if a protein interacts with a drug, it tends to change its thermal stability. And this was actually the initial purpose of developing TPP, was to find drug targets. But by now we know that it's not only drugs that lead to changes in protein thermal stability, but also metabolite interactions with other proteins or with nucleic acids, or post-translation modifications can lead to changes to protein thermal stability. Now, because we can monitor this plethora of changes in the cell, we wondered if we could combine TPP with a systems biology approach by perturbing the cell in many ways to start mapping the functional proteome, so characterizing protein function and mapping the protein interactions in a cell. So to do this, we selected 121 knockout mutants of E. coli. These mutants are involved in virtually every cellular process of the cell, perhaps with some enrichment of cell wall genes, which are important antibiotic targets, genes of a known function that we want to characterize further, and perhaps a depletion of essential genes, such as those involved in transcription and translation, simply because we do not have the knockouts to study them. Now, we took 2 independent clones of each one of these 121 knockouts. We grew the cells to exponential phase, and then we do our TPP protocol, which consists basically on heating the cells for a very short period of time just to induce protein denaturation. We lyse the cells when you collect the soluble protein fraction at every temperature. Then we take these thousands of samples, and then we use mass spec-based quantitative proteomics to quantify the levels of soluble fraction in each one of these samples. Now, before I get into the results, let me just show you what might happen to a protein in a certain mutant. So in most cases, most proteins are just going to behave like the wild type. But when you knock out a gene, some proteins in the proteome might get thermally destabilized or thermally stabilized. Because the cell is alive, it can up or down regulate genes to react to the lack of a certain gene. And of course, we can have all sorts of mixed effects. So what we do is for every mutant and every protein, we basically calculate 2 scores. One of them is the abundance score, which corresponds to the fold change at the 2 lowest temperatures. We know that at this temperature, no protein has started to melt. So any change at these temperatures must be a consequence of changes to the levels of the protein. And then we calculate the thermal stability score by correcting for any abundance change and calculating the area between these 2 curves. We take all the abundance and all the thermal stability scores, we normalize them across all mutants with a set transformation, and then we calculate significance from the 2 replicates using lemma, which is a robust t-test. So what we end up with is a heat map like this, where basically every column here is 1 mutant and every row is 1 protein profiled either in terms of its abundance or in terms of its thermal stability. So just by looking at the heat map, you can see that using thermal stability, we're accessing a completely different region of the proteome than if we were to do this exercise with abundance, which is what traditional proteomics does. We can zoom in on certain areas of this heat map and start learning more about mutant biology. So for example, if we zoom into this area, we have 3 mutants of iron sulfur cluster biosynthesis, and we have a large number of proteins that gets thermally destabilized. Now, a closer look at these proteins tells us that the majority of them includes an iron sulfur cluster in them, which makes sense. So in the absence of the cofactor, because the mutants cannot synthesize it, the proteins get thermally destabilized. And we can go one step further and look at all the iron sulfur cluster binding proteins in these mutants, and we see that overall they tend to be thermally destabilized. Now we can spend probably weeks learning something about all of these mutants, but we wanted to take a more global view and see if we could also learn something about all the other proteins that we quantify. And the first thing we did is to study essential proteins, which are typically difficult to study by genetic approaches because we cannot knock them out. And what we saw is that we virtually saw no difference between essential and non-essential proteins in terms of how they change across mutants in terms of abundance or thermal stability. But if we separate this in terms of abundance, we see that essential proteins change very rarely in their levels, which together with the fact that they are expressed at higher levels is consistent with the housekeeping gene. So they're or the housekeeping rule. So they're just expressed all the time at high levels, and their levels are never regulated. However, if we look at it in terms of protein thermal stability, we actually see that essential proteins tend to be regulated more commonly than non-essential proteins. So it seems that the cell has a way to regulate the activity of this protein at some post-transcriptional level. So to study this a bit further, we try to knock down a couple of essential genes using that Cas9 version, and we did this in diverse genetic backgrounds. So I'll show you one example today, which is FTSK, which is a protein involved in cell division. So when we knock down the levels of this protein in wild-type cells, you can see that the cells can cope with a certain downregulation of the gene compared to the control. However, if we do the same exercise in a mutant that is where this protein is massively thermally destabilized, you can see that the cells can no longer tolerate this downregulation. Importantly, if we do this in other mutants where the protein is not affected, we see that the cells behave like the wild type, perhaps with the exception of this gene MEA, which is also a gene involved in cell division. So there might be a different genetic interaction going on there. So this tells us that by using TPP, we now have a way to probe for this potential synthetic lethality between essential genes and non-essential genes in the cell. Taking an even more global view of the data, we can ask, are there pairs of proteins that change in a similar manner across all the perturbations? So an example here are two subunits of the RNA polymerase of E. coli, RPOB and RPOC. And I'm plotting here basically every mutant profile at a certain temperature. And what you can see is that there's a very high correlation between these two proteins. So as soon as one changes in abundance or thermal stability, the other one changes together very tightly. Now, these are two members of the same protein complex, but we can now ask a question for all protein pairs that we can detect. Are proteins that are highly correlated indeed functionally related, being part of the same complex or other functional units of the cell? And indeed they are. So they are not only part of the same complex, but also part of the same operon or part of the same metabolic pathway. So this is the type of enrichments that we see. And we can even go one step further and start to learn something about specific complexes or metabolic pathways. And today I'll just focus on metabolic pathways. So if we take central carbon metabolism as an example, and we just take all the enzymes that participate in the reactions across the pathway, we see that proteins correlate more tightly, proteins that are correlating more tightly also tend to be more closely related in the pathway. So this green cluster here are proteins that are part of the TCA cycle, while this yellow-orange cluster are proteins part of the glycolysis. And then we have this interesting cluster here in the middle, which are proteins that are part either of the glyoxylate shunt or side reactions or reversible reactions across the pathway that might not be active in all the mutants. So to study this a bit further and to understand if the changes that we're seeing are caused by protein thermals, are caused by changes of the metabolites in these mutants, we turned to metabolomics and we profiled 19 mutants and the wild type as a control. And we profiled with targeted metabolomics 6 metabolites of this pathway. And I'll show you one example here, which is succinate. And basically, we try to compare succinate levels with the thermal stability of SUCD, which is an enzyme that produces it. So every dot here is one mutant. And you can see, for example, in this mutant, succinate levels are low compared to wild type. And SUCD also appears to be thermally destabilized, probably because the metabolite is not there to thermally stabilize it. So overall, you can see that we see a good correlation between metabolite levels and the thermal stability of the protein, as we would expect. And we see similar things if you look at an enzyme on the other side of the reaction, SDHD. In this case, we see an anti-correlation, but still a very strong correlation between these two metrics. Now, interestingly, if we do the same in terms of protein abundance for the same two enzymes, we see virtually no correlation between metabolite levels and the levels of the enzymes. We can take a more global approach and look at all the metabolites that we have quantified across the pathway and the enzymes that bind them, and the picture is virtually the same. So we see a higher correlation between metabolite levels and the thermal stability of the enzymes that bind them than we see for protein abundance, which tells us that by using TPP, we have a way of monitoring a metabolic state of the cell. And importantly, we can do this in vivo because we can do TPP with living cells. And finally, we decided to combine our data with previously acquired growth phenotypes. So these mutants have been profiled across hundreds of chemical and environmental perturbations. And the first thing we see is we see a correlation between how perturbed the proteome is. So if a mutant leads to a high number of proteins changing in abundance or thermal stability, it also tends to lead to a higher number of growth phenotypes. So if the proteome is already perturbed to a large level, any external perturbation tends to lead to a growth phenotype. And we can go one step further and we can ask if we can explain growth phenotypes by proteome changes. So one example is by correlating protein levels to certain phenotypes. So here I'm plotting the abundance of a protein REC-R to sensitivity to UV, and you can see that there's a good correlation between the levels of REC-R and the sensitivity of cells to UV, probably driven by this one mutant, but this is simply because we only profiled 121 mutants, so we didn't populate this region of the plot. So the question now is this mutant sensitive to UV because it lacks this gene YBAB or because it lacks REC-R, which is DNA repair protein? If we, I just show you the same phenotype. So if we have the deletion of YBAB and we expose cells to UV, you can see that the cells are more sensitive compared to wild type. But if we overexpress REC-R, we can rescue this phenotype. But importantly, the same is not true if we just overexpress YBAB. So it seems that it is indeed the lack of REC-R in this mutant that causes the sensitivity. And if we didn't determine the proteome, we would associate the YBAB deletion to the sensitivity to UV. So just to summarize, TPP is a powerful new tool to map protein interactions and state in vivo. We are able, by using a systems biology approach, so perturbing and then a biochemical readout like TPP, to characterize functional units of the cell. And then we can combine our data with other omics approaches, such as chemical genetics or metabolomics, to try to understand the molecular mechanisms beyond the changes in metabolites or growth phenotypes. And of course, we did this in E. coli, but the approach is readily applicable to other species, and we are not limited to genetic perturbations, but we also can do chemical perturbations. So just to finalize, I'll just like to thank both labs where this work was done, thank my funding, and I'm happy to take any questions if there is time. Otherwise, just drop me an email or contact me on Twitter.
Thank you very much. Luckily, we do have a couple of minutes for questions. Well, I will take the liberty and just start. So is there, when you use the TPP, is there any difference on the readout if the protein is a substrate of any of the chaperones, for example, DNA-K or GRO-ELS?
I'm not sure we have looked at this in particular, but I would say that probably not. So one thing we have to keep in mind here is that the heat treatment is a very short heat treatment, so we are not letting cells respond to the heat stress, right? So it's just, we just want to induce protein denaturation. We don't really see that proteins that are less thermally stable tend to be chaperones or substrates for chaperones, I think.